I wasn't recording when I said that, and now no one can prove it.
Look at that.
Mr. Neufeld?
Mr. Newell, I will sit here.
Come get the notes.
Ooh, keep
I
calm.
No.
really enjoy my 2-day weekend.
If you weren't here last day, you need one of these.
I really enjoyed my 2-day weekend.
Don't worry that it's so thick. It's only part one. But it has lots of pictures and it's actually not that much. Anybody else?
Yeah. Can I get one?
You weren't here last day.
Well, you don't feel like it.
No, you need that. You need that here. Oh, there you go. Nicely done. Look at you. Oh, very good. Okay. I am really regretting buying that pencil sharpener.
It's very loud.
It is very loud. Okay, we're going to start biology. I'm going to give you some time at the end of class to do your lab. How many of you did not finish the test tube pouring reagents part of the lab and are coming tonight?
I don't know. Yeah,
Oh, I haven't
I
done
did
any of them.
something.
Did you sign up?
Yes.
Okay, did you sign up?
Yes.
Okay. There was a sign up. If you haven't signed up, if you didn't finish the actual lab part of the lab out in the lab, you will be having a quiz on Thursday. So you're gonna get your lab book at the back at the end. You're gonna keep the data sheet and procedure in it. You're gonna have all your observations. You're gonna answer all the questions in the back. You can ask me questions at the end. And then the quiz is open the lab book. So you have your lab book, you have your observations, you know what you're doing, you're good to go. Now, if you didn't finish the lab, then it is, of course, very important that you do finish it. So I have it open tonight, starting at 7:30. If you didn't see that, you didn't need to sign up, go through your email and find it, or come and see me, so I know you're coming. And I can email your host rank, or whoever's on duty. As well, as you may notice, I am ill, so I'm not going to write on the board. I am going to use notes that are pre-written so I can just talk. If I go too fast, slow me down and say I don't have all that down yet. And if my analogies seem off, I think I improved my analogy from yesterday, because yesterday's was a little wild. Keep calm and learning ATCGs. ATCG refers to the code in DNA. A always points to T, C always points to G, and it goes down in a pattern like that so that you can actually put that code into your DNA, and that will program everything that makes up you, and makes every single enzyme. The enzyme is the catalyst for every single biological reaction in your body, pretty much, which means if you control the enzymes, you control what comes out, and that is how we make you. So the first part is all about DNA. I used to say it's going to be a very interesting world because CRISPR's coming. And CRISPR coming is a bit like when we have supercomputers and saying it's going to be a very interesting world because the internet's coming, and everybody like, what are you with supercomputers? What are you talking? No one would have predicted the internet. We don't know exactly what CRISPR is going to do, but it's going to be very big. And I can now say CRISPR isn't coming. CRISPR is here. As of this year, we have reprogrammed the genes of someone who had sickle cell anemia using a human cure in order to treat their disease. CRISPR is a mechanism that we borrowed from viruses that insert DNA into precise points in your genome, which means we can precisely change what genes you have.
So you can get taller or what?
Yeah, I mean, theoretically, if you have genes that, if you could find genes that would make you taller, and then you injected them into someone who was young enough, at the right point of development, you could theoretically control how tall they got. Put drops in someone's eyes, and they would change their eye color.
Really?
Sickle cell anemia is what we would call low-hanging fruit in terms of the ease. It is a single point mutation, so it's very easy. If you're trying to cure stuff, that's an easy one to try and do because there's nothing complex about that. But if you're like, I want people who run fast, so I take the genes of a cheetah, because you've watched too many Saturday morning cartoons, and I will inject them into a person, and you get a person with like weird floppy cheetah legs, and they roll around the ground and they don't run anywhere because they're made for a cheetah, not a massive human. However, if you went the fast twitch, slow twitch muscle ratio in humans is controlled genetically, and we're going to increase the fast twitch muscle in our children because I want world-class soccer players. And then, yeah, you might have children that are slightly more athletic in certain ways. However, they'll be very bad at weightlifting.
Wow.
If you have slow twitch muscles, you can have stronger muscles with less mass. So it's the ratio. It's important. Most people have a mix. Some people are on the extremes. Having a mix is good for everyday activity. If you want to be the best in the world, however, sometimes the extremes are good. There is a family that has a gene that gives them an extremely large amount of their ratio of slow twitch to fast twitch muscles of slow twitch. And they build muscle without having to exercise. They just get massive muscles and they don't have to do anything. They have more medals for weightlifting than any other nation in the world as a family. They also have a lot of people in their family who are restricted to wheelchairs and can't get around because that particular gene is in most people deleterious. Your legs don't develop properly and then you can't walk. So some people it didn't affect them enough and they just became massively strong. So if you went and got, yes, CRISPR, I'm going to get that gene, most of the people you experiment on are probably not going to turn out positive. So
Wait. So then, what if it did? You just like.
yeah, I mean, if you don't have ethics, you just keep doing it until you get one you want, and then keep that one.
Isn't that just eugenics but different?
Yeah, it's eugenics with extra steps. Eugenics
So
is
it's
just to be
bad.
clear, so that everybody is aware. Eugenics is bad. Then now that I see someone texting and I see that there's only one phone over there, can everybody please take their phone and put it over there? Yes. Yes.
Eugenics is bad.
Eugenics is bad.
Controlling, trying
Well-
to make better humans through
To be very clear,
breeding.
having other people control genetics is eugenics. So if you have the nation deciding who's going to live and who's going to die and produce babies, that's eugenics. If you yourself want to cure your child, that's just called medicine. So eugenics is when it's applied from the outside in a broad scale. That would be bad. CRISPR then has the potential, if you have one of those very common form of diabetes, it's a single point mutation, theoretically now cured. You have phenylketonuria, you can't digest certain types of protein, you have a special diet for the rest of your life. Your life is made much more difficult, theoretically cured. There's all these diseases now that are fairly simple genetically, but we haven't had a wrench small enough. If this first one works, we have a wrench small enough and we can now start curing these things. But it also leads to other things. Your eye color, is controlled by a complex number of genes, but theoretically, change it with a drop. You could get a gender change for your birthday because XY versus XX chromosomes, and then just switch back on your next birthday. And then
Very great.
design a virus with CRISPR on it and deliver genes to anybody you want, infect the population. Maybe you really like glow-in-the-dark skin and you want everybody to have it, and you implant the luciferase genes, not in people who want it in the form of glow-in-the-dark tattoos, which could theoretically be possible, but just everybody for fun, as a prank. There's all sorts of problems that may come about, or may never happen, because it's not technically possible. Right now we're just looking at the fact that we can do it, but it means that we can do a lot more than we used to be able to do. Genetic therapy is old. Back in the 80s, we were trying to cure the severe immune deficiency disease. There's a film once made, Boy in a Bubble, that was a comedy, not a very funny one. But Boy in the Bubble disease is you're stuck in a plastic bubble your entire life because if you get sick with a virus or bacteria, you can't fight it off. You have no immune system, essentially. You live in a sterile room. Everything is delivered to you through a a double-doored little passageway thing that they can put things through and make sure it's sterile before it's put there. Your parents either have to wash up really well before they give you a hug or hug you through plastic, because if you get anything from anybody else, you'll die. 1 point mutation causes a cascading failure in the immune system, and some people have this. There was a young man who was going to turn 18 in France, and he said, I don't care if I have this particular disease. I am going to come out of my bubble as soon as I'm legally allowed to, and my parents aren't in control of my life. And if I live a month, great. If I live 2 months, fantastic. But at least I will be living and not stuck in a room for the rest of my life.
What happened with him?
A bunch of scientists said, hey, we're doing this study, and we're trying to cure this with genetic therapy. So genetic therapy is old. We've been trying to do it for a really long time. Would you like to sign on? And the young man said, yeah, absolutely, sign me up. So he lived for 2 years, and he died of leukemia. Leukemia is not a transmissible disease. He did not die of a virus or of a bacteria. In fact, those 2 years, he was just as healthy as anybody else. He was cured. The problem was, is if you inject things into your DNA, you're creating a mutation. And it happened to be injected in a growth area for blood. So that gave him eventually cancer, because cancer is due to mutations. So our cure did put in genes, but it also had a very high risk of giving you cancer. Out of the 8 people they did human trials with for that test, 6 of them died within 5 years. The other 2 are alive today and are doing perfectly well. They are completely cured. So it worked. It's just that 2 out of 8 ratio is not considered good enough for ongoing medical therapy. What CRISPR does is it takes that random event that makes a mutation and it allows you to say, put it here and put it exactly here. And so it takes the randomness out of it and makes that risk of cancer go way, way down. So this is going to be an extremely interesting time.
Are they ever going to figure out how to cure cancer or not?
Cancer is not one disease. Part of the problem is that we call cancer a single disease, and it's not. It is a bunch of different diseases, each of which requires its own curative therapy. So, would they cure a particular type of cancer? Maybe. It is a mutation. CRISPR has the potential, if you could rapidly sequence the part of the cancer that's a problem, and then develop a therapy from that sequence to attack it, maybe. Part of the problem with cancer is it's part of you, and so it looks like you. It's hard to attack something that looks like you genetically.
And everyone's cancer is different.
Yeah. So something like CRISPR, if we can process the information fast enough, like get a sample and do it, maybe. There's people are very excited about CRISPR for that, but that's not low hanging fruit. That's high up fruit in the tree. It's going to be a little while before we do that. Okay, next page. History of DNA research. DNA, now this is not a history memorize the dates course, but I want you to have context of time, so that's why I put in 1869. He was studying pus in medical bandages and managed to extract DNA. So he discovered deoxyribonucleic acid as a compound that was in living things by studying pus in medical bandages. He didn't know what it was for, he didn't know its purpose, he just knew it existed. 1869. Nobody knew what it was for. A little while later, we name it DNA, deoxyribo. That is wrong. Sorry, that must have been the text recognition going wrong. Deoxyribo. I'll put spaces. It's all one word, but it is not. The spaces aren't related. Deoxyribonucleic acid, DNA. Deoxyribose is the sugar, which is where the ribo comes from. Any ose you see as a name is usually a sugar. Ribose is in here. Table sugar is
Glucose.
glucose is blood sugar. Sucrose is table sugar. Fructose is fruit sugar. Pardon? Corn syrup is, people will complain about high fructose corn syrup. Fructose is just the sugar you find in fruit.
So,
And when they take corn,
which is
and they chemically process it to turn it into sugar, it ends up having a high amount of fructose. Lots of things have high amounts of fructose. There is some indication that high fructose sugar might be a little worse for you than other types of sugar, but the main problem is that high amounts of sugar is generally not good for you because you are built to eat sugar with food that is good for you. If you were running around on the plains of Africa hunting gazelle, as you were born to do, what kind of things would you find sugar in?
Fruit.
Fruit. Basically just fruit. And maybe a little bit in a root here and there. And so sugar evolved to let you know that there was lots of nutrients in this, and sugar itself is high energy. It's really useful if you're literally running around after gazelle. So you need that energy. It tells you this is a great food to eat lots of. Cleverly, we then extracted the sugar from the vitamins and the fiber and everything else, and then made chocolate bars and gummy bears out of it, so you don't get any of the nutrients, you just get the sugar. And it turns out that if you purify it and give it to people in straight amounts, it's not that great for you. Sugar's fine, eat it in fruit. If you're worrying about high fructose and low fructose syrups, mostly you should just be worried about syrups. Eat the fruit. Candy's not bad, but it's bad every day. What are you looking for?
What's the word after cause?
Bacteria! Obviously. Obviously. Let's see what it says. Nope, it did not like that.
What did it think it said?
Some
Oh
version
my god.
of a curse from Harry Potter. Let's go back. How did it get that out of it?
The T looks like an A.
There, that one's a bit better. I am going to go over this experiment in detail. It's a very simple experiment, but I find it takes a little bit to understand what they did. And it's an amazingly clever way to figure out that DNA was the cause and was carrying some sort of information. So, also keep in mind, first discovered, 1869. Here we are, a little over 100 years later, and we're just trying to figure out just starting to figure out what it does. The behavior that it changed is it made bacteria go from not deadly to deadly. And so that's what they're looking at. It allowed viruses to infect cells, indicated it's played a special role in living organisms. I also have to talk a little bit about the discovery. I made sure that the picture that you see is not Watson and Crick, because Rosalind Franklin was instrumental in the discovery of DNA. She was not credited in the original paper, she was not acknowledged, and she had her photographs stolen by Watson and Crick. Watson and Crick figured out the structure of DNA. They were given these models. In here, you can see that there's a ring structure there. Let me just draw it out a little bit because it's probably kind of hard to see where you are. Maybe a different color. And so they had these models, and they inherited these models. And by the way, these are just ring stands like you use out there. So they're literally in a chemistry word drab, just using ring stands and little models that they've made out of bits and pieces. They inherited them from Dr. Linus Pauling. Dr. Linus Pauling is a very famous chemist. He had won 2 unshared Nobel Prizes, the only person ever to do so, one in chemistry for his work in electronegativity, and the other in peace, because he went around the world as a diplomat. He was such a famous chemist and did a ton of work negotiating ceasefires and peace in the Middle East at the time. He was also working on the structure of DNA, and he made these models of the chemical compounds, but he couldn't figure out how they quite put together. Eventually, he got frustrated, and he put them aside, and he was like, I'm going to maybe come back to that later. I can't do this anymore. And Watson and Crick said, we're trying to figure out the structure. Can we borrow your models? And science, being what it is, it's very open about research and just wants, the answer. We don't hide things. Linus Pauling was absolutely, take the models and work. So very generously, they started putting together the structure, and they realized they had no idea. They were working on it, they couldn't figure out how to do it. Rosalind Franklin was also working on the structure of DNA, and she wasn't working with models, she was doing X-ray chromatography. And to explain why would take a bit more time in terms of what does this mean. But this picture tells you that the structure of DNA is a double helix. So having that is having the cheat sheet. You look at that and you go, oh, now I know how it's put together, and you can go and now figure it out properly. It's like getting the picture to the puzzle box. You haven't built the puzzle yet, but you've gotten 90% of the way there if you have the picture first. They literally stole that out of her lab. They didn't give her credit on the report. The first time she saw her picture in the paper was when it was published. They haven't retroactively awarded her a Nobel Prize. The Nobel Prize Committee says because they don't award Nobel Prizes in retrospect to people who aren't alive. You have to be alive to receive a Nobel Prize, so they're not going to correct that as their policy.
Did they
Pardon?
get a Nobel Prize?
Those two? Yeah, they won a Nobel Prize for the structure of DNA.
They should get approved.
Yeah. Well, they did discover the structure of DNA with someone else after getting the models from someone else openly and then didn't credit her. It seems sad. I have an image too because X-ray chromatography takes tiny pieces of the molecule. So you literally take DNA and you make a little, it's really stretchy like an elastic band when you have a chunk of it, pure. And so she'd have to stretch it between two thin needles and then face the X-ray chromatography machine at it. And because it was like stretchy, it would spring off half the time, boing, and fall on the ground. So I have it, and then she would crawl around on her hands and knees looking for it so she could put it back on, because it took a while to make the sample. So I have an image of her crawling around on her hands and knees looking for the DNA that sprung off. Meanwhile, Watson and Crick are sneaking into the lab to grab her picture. However, they worked there, so they probably just took their keys and snuck in when she wasn't there, practically. But absolutely, she should have been on the paper, and it's a crime that her name isn't as famous as Watson and Crick.
So the guy's just like,
Straight up stole it. Yeah.
The tale as old as time.
Yeah. Now, did she come up with the total structure? No. It absolutely was. I don't want to say that Watson and Crick did nothing. They were absolutely instrumental in discovering the structure of DNA. But it's also important not to lionize people before their discoveries. You can be an incredibly bad person and still be right. They happen to be right. But they stole it, which wasn't cool. Okay. I am going to go through this one experiment at a time. I'll go over the messy writing. I made an analogy to make it a little easier to understand that I found. I'm going to change the analogy slightly because in my sick state yesterday, I made it a little, I think, too violent. And I'd like to point out that any analogy or anybody listening from my analogies from yesterday, everything was paintballs. Sure it was. Okay, you have a rough strain of bacteria. So when we're talking about rough strain, we are talking about a type of bacteria. The word non-virulent means not lethal. That's an A. What they did is they took that rough strain, and this is a control, so that you're just doing this to show results you know, but to show any potential change that you do. So you take the non-lethal bacteria, you inject it into a mouse, the mouse lives, which makes sense. It's non-lethal, that should happen. My analogy is, no, I'll do the analogy after. You take what's called the smooth strain, which is also bacteria. This one's virulent, and that means it's lethal. Inject that into the mouse, and the mouse dies, which is an expected outcome. So my analogy, the mouse is this Science 10 class. The mouse is this science 10 class. The grade 8s burst in with paintball guns. And what happens? We take their paintball guns away from them and we shoot them. All the grade 8s die. We're fine. Mouse lives. Or, in this case, the smooth strain is Joint Task Force 2, the Canadian equivalent of the Special Forces, burst in with paintball guns, and we all die right away. I found out in researching, because I was like, who is Canadian Special Forces for this analogy? It turns out Joint Task Force 2 recently won 40 American military medals. And we don't know why, as the Canadian public. They just won them, but everything about it is classified. Apparently, they're very good. Yeah.
Always snatch you up in a second.
Yeah. Okay, next one. This is where the analogy kind of goes off a bit. Anyway, so we take our smooth strain bacteria and we heat it up. Microwave, autoclave, oven, don't care how. We heat them up until they die. Every single one of them. We inject it into the mouse and the mouse lives. That makes sense. Dead bacteria can't infect the mouse, doesn't do anything. We take Joint Task Force 2 and heat them up until they're all unconscious, just so no one dies. And then wheel them in here with all their equipment. We're fine. Yeah. We just defeated Joint Task Force 2. Well done. Okay? So, alive bacteria, dead bacteria. Dead bacteria don't cause the mouse to die. Good? Excellent. We take the dead, smooth strain bacteria that's deadly, the dead ones, and mix it with the rough, non-lethal ones that are alive. And we inject them into the mouse, and the mouse dies. Neither of these should have killed the mouse on their own. So this would be the equivalent of, we take the bodies of Joint Task Force 2 that are all unconscious, and we take the alive and moving grade 8s and mix them up together, and they burst in here, and we all get shot. What happened? How did that happen? Yeah? Yeah, they have way better equipment. They probably even have written battle plans. They take it and in their incompetent way manage to take out our Science 10 class. Grade 8s are fantastic, apparently. So, what must have happened between the bacteria? What? They took. Well, we don't know about genes yet. They took something. They took their weapons. They took their battle plans. They took something from the heat killed. At this point in the experiment, we don't know what, but now we know there's something that can transfer that gives the rough bacteria the weapons they need to take out the mouse. Yeah. And then there's only one more experiment, but that's a bit of a lie because there's actually a bunch of other experiments that happened. Because what they would then do is they'd go in and to the dead smooth strain, they would like chop up all the proteins with an enzyme. They would chop up all the ribosomes with a enzyme. They would destroy various parts and see whether it would still work. And the one that worked was when they went in, they chopped up all the DNA. When they chopped up all the DNA, the mouse lived. So this would be the equivalent of going in to Joint Task Force 2, and when we chopped up all their weapons, the grade 8s all of a sudden were incompetent again. Turns out they had better weapons. So now we knew from this one, it was the DNA that was being transferred. That's what gave the information or whatever it was to the rough bacteria to make it deadly. A really clever set of experiments. And that's a nice guy.
That feels like 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 when they were testing like every enzyme, just being this is mouse number 32.
Oh, it's literally like that.
I mean, like the DNA mouse, the last one left in the cage, looks up, a hand reaches down.
Okay, let's not dwell too much on that. Although I run not with mice, but chemical experiments where you do a control, like this happens and then it happens, and then you have, oh, okay, I changed this one thing. What happened? I changed this one thing. So I've had that where there's like literally like 40 vials where I'm changing all of them just to find the one where I make one change and it does it. You can't make a bunch of changes because then you don't know which one it was. So you only can change one thing slightly at a time.
Do an experiment like this these days, everyone would get mad at you.
Oh, not only do we do experiments like these nowadays, we have mice that are specially bred to grow specific types of cancer so we can figure out how to cure them.
Oh,
Yeah.
poor mice.
If you break into the UBC laboratory, as some protesters did one day, and release the animals, you have given some of them a very, very painful outcome. Some of them, for example, have been bred to require certain food that's metabolic,
Yeah.
that must be given in a lab that does not exist in a natural form in nature to study metabolic pathways. So if you free it, you have just condemned it to a life of starvation. It's not nice. If you're going to free the animals and do against animal testing, the makeup and moisturizer and shampoo and cosmetics industry does a ton of animal testing that is not only unnecessary from a scientific standpoint, doesn't actually even, it's not even good experiments. They're doing it for legal reasons just so they can say they did it in case they get sued. But if you've done it once, you don't need to do it again. And a lot of their studies, when looked at by scientists who study things, as opposed to people who are doing it for legal reasons, are like, that doesn't even prove what you said it proved. And that is by far the bulk of animal testing. The ones we do in the lab for cures are actually a very small part of all animal testing. Not that I'm encouraging you to break the law and break out the animals that are involved in the cosmetics industry animal testing, but they're not even scientific necessity.
Wait, as in they freed the animals, so they just like break their cages and that?
Yeah, and then run them out, run around
In the
Vancouver.
And then they went and died.
They open up the doors.
Thanks,
Okay.
minions.
Like this happened a number of years ago, and they opened up the doors
No.
and let them
So
go.
looking at the ceiling doesn't make it unclear. And your childish comments are
Well,
funny.
Yes.
If they definitely knew that the lethal one is gonna kill the mouse, like is it like like why would they even
It's a control.
It's a control.
It's a control to show that this strain that you're using, because strains also mutate. So if it's not needed,
would they choose to kill it?
if there's a way, like you're using a strain that's well established and they know what to do, they may not actually run that control. To do any experiment involving animals in the scientific community, you have to go before an ethics panel at your university and have it approved. And a lot of them are rejected. One, hilariously, a psychology professor wanted to study stress. And so what he wanted to study is he regularly, in his class, gave surprise quizzes. So he wanted to study with volunteers from his class. He wasn't going to make them, but anybody who volunteered, the effect of that stress on their exam performance and what chemicals are given off, and then he could look at a whole bunch of things. And the ethics committee said no, because the amount of stress that he gave, that a surprise quiz was considered too horrendous a punishment for students to do as an experiment. So he's allowed to do it as a teacher, but the ethics board won't let him do it as a scientist. We have a history as humans of really bad medical experiments in which we tortured humans. I used to teach a course called Science and Ethics, and we would do a whole little project on it, and I stopped because it was so depressing. If you want to look up unit, was it 731, some of the testing in some of the medical testing in World War II was just awful. And then in the States, there was post-World War II, for example, they gave a bunch of prisoners syphilis when it was curable. They didn't cure it. They just gave them syphilis just to see what would happen for a variety of reasons. Because they were prisoners and their skin color was the kind they were worried about. So the history of racism and human testing means that if you're going to do a test on any animal, humans or not, it goes through a lot of processes. So I was involved, I'll do it after, in animal testing. I'll get your question next. At the University of British Columbia, they were trying to cure Parkinson's and Alzheimer's. And they're going to do that by taking some cells that they reverted back into a state that they could become any other kind of cell. And then inject that into the brain of a human and have it grow and replace the part of the brain that wasn't producing the right type of chemical compound. Because I worked for the Tri University Nizon facility, and they were doing PET scanning. You could actually tell people, they would get the brain scanned, and a tiny area of the brain would start to go dark. That meant it wasn't producing enough of that chemical. Even before they showed symptoms, they were able to diagnose people with Parkinson's or Alzheimer's. They did that on a few occasions, people who just volunteered as normal subjects, because they needed a lot of volunteers for normal subjects. And occasionally be like, sorry to tell you this, but by our results, it looks like you have early onset Parkinson's. So what they were doing is they were inventing the cure. There is no way to test this in a test tube or a petri dish. This involves a brain. You can't use a human brain, so what brain do they want to use? Monkey brain. Chimpanzees are the nearest, and apes are the nearest analog to humans. So they gave the apes Parkinson's disease first, and then they attempted to cure them with this method.
How do you give Parkinson's?
How do you give Parkinson's disease? Well, if you were in New York in the 1970s, there was a really bad batch of artificial heroin that was apparently going around. Someone tried to make synthetically heroin in a lab. And all these young adults, like 20, 21, 17, were showing up to emergencies with Parkinson's disease. And the emergency room was like, what is going on? Is this now a transmissible virus disease? Like, what's happening? And it turns out, no, the heroin was destroying a part of the brain. It wasn't heroin. It was another chemical they accidentally made that just gives you Parkinson's. So now you have a chemical that can give someone Parkinson's.
Oh, that's
So they gave
great.
monkeys Parkinson's.
Parkinson'sizer.
And if they cured the monkey, guess what happened to the monkey?
We killed it.
They would euthanize it and then carefully slice his brain so they could see exactly how it was cured. That had to go through multiple ethical reviews before they could do it. Not only are you using an animal, you're using a chimpanzee, which could be argued has quality of life, has thoughts, feelings potentially, like there's all sorts. So like that's the most extreme. And the stakes are, can we cure Parkinson's disease? So you have to have those kinds of stakes to argue it. You can't just do it for fun.
So they end up like making it actual like.
It gave them important insight into why that method wouldn't work, and they stopped 2 years later. Yeah, the particular types of cells they were using weren't able to grow in the right way. So, like most science, what they got was a useful negative result that will never be published. I volunteered to have my brain scanned like that, because I was working there as a chemist. And then I went through the process, and they said, actually, by the time the summer's over, I had to go through so much testing in order to be able to scan my brain. The summer would have been over before I could have it scanned. I'd be back at school, so they said no.
Mr. Nizam,
I was so excited to be injected with radiation. Yes.
if they didn't know that DNA caused the different traits, how could they identify strains before that? Were they just
Rough and smooth literally refers to their appearance. So when we call it a rough strain, we mean that when you grew it in a petri dish, it was rough looking. And when you grew the other one, it looks smooth and round and kind of glistening.
Okay, that seems a little
Yeah.
bit dumb.
We also have gram-positive and gram-negative, which just refers to a test, Graham's test, whether it's positive or negative. And that tells you a little bit about the bacterial cell wall. But you don't have to know what it is. It's just the result of a test or how it looks. Okay, DNA is an extremely large molecule containing millions of atoms joined by chemical bonds. If you took the DNA out of a single cell and I unraveled it into its one long line and I put it down, it would be 2 meters long in every single cell.
How many cells are there?
Well, if you took the DNA out of every single cell and lined it up, you would die.
Well, no, really?
Yeah, because you don't have any DNA in your cells. I don't know, some imaginable distance, like go to the moon 2 or 3 times. I don't know, that's just as incomprehensible as anything else. But I find the 2-meter one to be somewhat useful.
I want to hear the song.
So it's extremely large, it's got millions of atoms. How many of you have seen The Big Bang Theory?
A little bit.
Yeah, it's not that popular anymore.
Oh.
They have a nice big model of DNA in it that's built on the floor like the models that I have here. It winds around. You can see the rough and smooth. I want to get one for this class for the science lab. It's only about $2,000 and about $1,000 for shipping, and then you have to build it once it gets here. And I of course have free labor.
I'd build it.
It would be so cool to build. I do have to convince Miss Allen, so this is a mandatory, you have to put up your hand for this question. How many of you think that the science department would be improved by getting this model? Everybody must put up their hand. You have to put up your hand. You have to put up your hand. You have to agree. There, excellent. Everybody in every one of my classes has agreed. Well done, thank you.
It wasn't forced at all.
Not at all.
I'm gonna tell Miss Allen. I'm just kidding.
I'll tell Miss Allen that we should get this model.
Okay. We're going to draw a nucleotide in both simple and complex, not super complex. To give you an idea of what one piece of DNA looks like, a nucleotide is one single piece of DNA. It's got bonded on both sides. We have here, that represents the phosphate group. What is phosphate from your memory as a polyatomic ion? PO4 minus 3. So this is not just a bowl. It's got like oxygens and all sorts of things on it. So that's the but in biology, the biologists don't always like drawing the exact chemical structures. We want to just give the overview. So they make a little circle. The phosphate, this, and it disturbs me, there's not an oxygen there, is the ribose sugar, the deoxyribose sugar. And then this is one of 4 bases. We usually make it a square or rectangle. It's a base. It's specifically a nitrogenous base. So this entire thing no hiccuping allowed. Is a nucleotide, a single nucleotide. You'll notice there's a pattern. Pardon? The little purple thing is the nucleotide on the other side. You'll notice they're also directional. On one side, the ribose sugars point up. The other side, they point down. So they do this. That's important in terms of how you read it. Do you notice a pattern in how they bond? A and T and G and C. So pointed and sharp, always. Sit up. Shake your head. Sit straight. There you go. The sharp ones, A is sharp, T is sharp, go together. The round ones, C is round, G is round, go together. They fit together because they have hydrogen bonding. You'll notice those dots. How much hydrogen bonding does C and G have? From the diagram in front of you. 3. A and T? 2. And that's why they don't fit. If a C tries to bond with a T, it has the wrong number and alignment of hydrogen bonding. And so they only fit in that particular way. Yeah. Let me delete all of that.
How did this come to be?
Write this down. We got each DNA strand is made from repeating molecular units called nucleotides. Chargaff, the 1950s, showed that they don't repeat. And what we mean by that is that it wasn't just a nonsensical pattern that was like A, T, C, G, A, T, C, G, A, T. It looked like a code because it seemed informational. It didn't repeat in a way that just a random process would make statistically an even number of everything. It looked more like there was some kind of information there. And so that was pretty interesting in terms of discovering, like, is this where we get our information from? Now, he also showed that the percent of A equals the percent of T, which it should, A bonds with T. So, as an exercise, if I have 22% T, how much C do I have? Any guesses?
28.
How did you get 28? I'm not saying you're right or wrong. I'm just saying, how'd you get 28?
Because it'd be 22 plus 22, 44 minus 100 minus 44 is 56, and half of 56 is 28.
So if this side is 22%, we know that side has to be 22%. But how much must it all add up to?
100.
100%. So each side must add up to 50. So that would be 28%. And that would be 28%. So if you knew the percentage of any one of them, you knew the percentage of the other one. Because it had to pair that way. This also told them how it paired. So given that you could predict the outcome, they knew that C always paired with G, and A always paired with T.
And this is an example. It's not actually 22%.
No, it may be any particular amount. It's just now. Yes.
When they didn't know that did they know it repeated when they first discovered it?
Oh, and when they first discovered it, they didn't know it didn't repeat. There was biochemically, there was a ton of argument over this. There was a lot of biochemists who were like, get lost with this nucleotide DNA thing. There's no way that's information. Look at the complexity of proteins. Proteins, you have like 20 different types of amino acids. You could pack so much information into that. What is this? This is like 4 letters. How is that going to code for anything? And they were like, and there was a strong biochemical argument for complexity that like this wasn't complex enough to be informational. So they had to prove it. This is one of the early ways they were like, there's something more going on here. Because these aren't, if it was just random, you'd expect it with that many to be 25, 25, 25, 25. Just like coin tosses eventually get to 50-50. Okay, next page. Last thing we're going to do. I'm going to draw this. You have nothing there, right? Yeah.
I have some.
I will delete this first so that you can see
Well, let me
everything.
copy it first.
Yeah, that's why I'm going to do that, and then I'll add those bits back. And this will be the last thing that we do. You've been very good. Today's been a pure lecture, mostly. Because at times I feel like I'm going to fall
Okay,
over.
time to draw this.
Now, the phosphate, PO4, true phosphate. So it's not just a circle. However, if you're a biologist and you want to draw it quickly, they tend to just draw it as a circle and put a P in it, and they say, that's phosphate. If you're a biology student in particular, they like doing that. Because I find historically that biology students went into biology because they like science, but maybe they didn't like chemistry.
clueless? Like,
In the same way that physics students go into physics because they like science, but chemistry was too hard and they just needed formulas.
Didn't you want to go into physics to begin with?
Yeah, but they all smelled.
And the
Weird.
chemists didn't?
Um,
Oh, the chemists made
Nick?
soap.
he is so out of it.
Nick.
Nick.
I think if doing the Mr. Snow thing where I walk up and slam a big book on the desk would probably be very frightening. But
Does he do that?
he did that once.
That
Yeah.
I've seen it.
sounds scary.
It wakes you up. Well, it does.
No, apparently he broke a table.
He broke a table?
Yeah, it was like the first week of school.
Ah. Everybody needs that. Nick, maybe you took your jacket off, made yourself a little uncomfortable. I can give you some tacks to put on your seat if you'd like. That's a joke. But I will say that I literally did that in university once to stay awake during a lecture. I had to study for a midterm and I was up more than 24 hours and then I had a lecture that I could not miss and I had to be there for and I was like falling asleep during the lecture. I pulled out tacks from my bag and put it on my seat. Thumbtacks.
Thumbtacks.
Sharp. Like you pin posters to a wall.
Oh,
Yeah.
what that? On your seat, so you see.
Yeah. I put them at the back so if I started to slump, I would get pain.
Oh.
Just sit there.
Really?
Fall asleep there.
Sometimes
Yeah, you wake
Sometimes
up
studying
and
chemistry is
your ass
hardcore.
bloody.
Here's our deoxyribose. It's deoxy because it doesn't have this. Oops. As a biochemist, it should have another OH, but it's a deoxyribose because it doesn't. Also, this is I'm actually embarrassed to say right now, I can't figure out which base that is. I used to have them all memorized. But this is one of the Stop. Pen green. There. This is one of the nitrogenous bases. We often will just draw it as a rectangle and we'll just put in whatever it is. A, T, C, or G. I am not going to ask you to memorize this entire structure, but I want you to draw it at least once so that you know the structure. Also to explain some biochemistry drawings. When you see a structure like this, these aren't random points. Like, why does it look that? Every one of these is a carbon. Because carbon is so ubiquitous and common in biological drawings, every time there's a point, there's a carbon. You don't have to draw the carbons in because it's accepted that you don't have to. And notice there's no hydrogens unless they want to show it off of something else. So carbon has 4 bonds. That means this carbon only has 2, so there's an H and there's an H, and that's true for all of them. This has 3 bonds, so we know that has 1. This carbon has 4 bonds, so no hydrogens. So you can mentally add in the hydrogen if you want, but to make it less complex and easier to see the structure, essentially biochemists just say, don't draw the carbons. We know they're carbons, just draw the shape and put the things in that aren't carbon and hydrogen.
Interesting things.
Yeah, the interesting bits, basically. So, what do I expect you to be able to draw? After you're done this, I would expect you to draw something like this. And put into it that, like, this is a phosphate, and label it. This is a ribose sugar. In fact, in most cases, if you just say it's a sugar, that would work. And you should be able to label that as either A, T, C, or G. And know where those bases are. It is also, you'll see it commonly referred to as a nitrogenous base because it contains a lot of nitrogen. So it's this particular type of base that doesn't have hydroxide that we teach you about in chemistry 12. Yes, and you don't have to ask. Don't go to sleep there.
I'm there, wake up.
He's gonna fall asleep on the floor.
Okay. I am going to stop there. You can now grab your lab book, do the questions. You're going to take your lab book away, leave in the marking sheet. I've had a look at them all. but I'm not gonna fully mark them or write on the sheet until I've seen both your quiz and any final things that you've done, okay? You'll get your lab book on your quiz, but it's still a quiz. It's not multi-person or internet enabled. You only get your lab book and potentially your data booklet. I'm going to do a correction on your lab book as lab procedure as well. So if you can go grab that, so you can look at that when I do it, and then you'll have time to work on the questions, and you'll have time to work tonight and prep.
Wait,
So grab your lab book.
what if you haven't completed the lab yet?
You're coming tonight.
Yeah, yeah.
Bring your lab book.
Oh, okay, so what do I do now?
Grab your lab book. It's where they were all left.
Okay, but
Yeah.
I don't do, what do I do this class?
You can leave yourself some space to do the questions. You can do the questions and then start a table as long as you label it properly. That's fine.
Well, no, I've done like half of the lab.
Yeah. You can work on the questions still, even if you're not done it. You have enough information.
Is it?
Sorry, are these them? Yeah, I moved them. Layla? Jamie? Is anybody in Jamie's house? Pardon? Oh, she's not gonna be back for Thursday? Yes, she will be back, or she then we might as well not have give it away then. Grab your lab books. Hey. What were they? What do you mean stop? Who was touching you? Who? Arnie, don't touch people. You need consent. Do I need to show the tea analogy?
But what?
The
Some
tea
people
analogy.
don't like tea.
Don't make people drink tea,
Don't be-
especially if they're
They're
listening.
unconscious. What's such?
What?
Don't force people to drink tea.
Okay, I've
was
never
still
seen
here.
that.
Great.
I've
Have you
wanted
seen the
to.
Viking? Have you seen the Viking? The Viking consent. Don't force people to bike.
Whose lab is this? It has no name. Beautifully written pre-lab right here.
Oh, that's
Um,
totally not.
It's in pencil though. However, it can always be fixed. And then nothing else. It might have been an emergency lab book. Nobody here.
Aruga, emergency
Okay.
lab.
And Grayson. Is anybody in Grayson's house?
I don't know. He wasn't in business.
No, but is anybody in his house? Clark.
He wasn't perfect.
Why are you laughing? Oh. Are you in his house?
No one here is from his house.
What house? He's in the Whittle. Nobody here's in the Whittle?
Yeah.
Nobody goes to Whittle. Whittle's a sad and lonely
Whittle
place.
isn't.
Whittle is, yeah, Whittle's
Whittle
not
isn't
real.
If there's no Whittle people here, I can finally say, someone I'm not going to name, when I first started working here, I talked about the houses, and I talked about Whittle House. And the whole time, for 2 years, they're a member of staff. They thought I was talking about Whittle boys. Because they're Whittle.
They are.
Also sad. And then I'm wondering, like, if they always thought that, what did they think Privet, Ellis, and Rogers were? Anyway. Okay, number one.
Number one.
Open up your data booklet to this page.
Liam,
Page 5.
Your grade just went down, Liam. Let's be honest.
No, I love my
Science.
data table, science, and everything.
Your lab, anything from the questions, your observations, or this table. Pardon? There's a whole backside with questions.
Oh, yeah,
Yeah. It's in your data booklet. Data booklet, not in the lab.
See if I can find a data booklet.
It's not funny.
If you need a data booklet or speed, I'll just give you one. Who needs a data booklet?
You got one.
You got one, right there.
Oh, well, he gave me another one.
Turn to page 5. You here. No, want another data booklet? Oh, then there you go. Data booklet. Your data booklet's on that. Get another one. Yeah. just throw out data booklets like candy.
Okay,
When I signed it,
Can I
You
have
can
some
always give
candy?
one back.
I want candy.
of course it is.
There you go.
no.
Okay.
No, I'm
Uh,
working. I already have one.
If I have, Aiden, if I have something that's basic, let's say ammonia, pH of 11, and I put it in phenolphthalein, what color does it turn? Yeah. Magenta, if you want to be classy. I always call it pink, because never quite sure what magenta is. But here it is, and you can use the chart to see for any indicator. So phenolphthalein, if you forget, is on your chart. So you can go, basic. Notice it doesn't turn until about a pH of 8. So neutral or slightly basic will be colorless. Litmus is great because it turns right on 7, red or blue. So that tells you if the pH is above 7, below 7, or equal, and if it doesn't change either way, equal to 7. But I can also ask you a question. I can say, I have soap, and I test it with indigo carmine. What color will it be? Blue. Here is soap. Here is indigo carmine on the bottom. I go down, it's blue. Indigo carmine just tells you if things are really basic. Essentially pH of 11 and a half about and above. If I said I am testing grapes and I'm testing it with methyl orange, what color would it be? Red. I'm testing eggs with bromothiamyl blue. Blue. So you should be able to use the chart to get the pH of anything. If I don't give you a data table on your quiz, I will include this on a separate page or something. In terms of speed of writing, you won't need your data table. I mean, I might give you a data table anyway. Depends if I'm there. I am feeling very ill, and I may be putting in for BFA tomorrow. I really don't want to on Thursday because that's the day everybody goes home, but we'll see.
It should be Mr. P.
Pardon?
It should be
Mr.
Mr.
Who?
Deline.
Deline. Second correction. If you could turn to your procedure, there is a mistake in the questions. Don't make any changes just yet. However, this is what was written originally. I turned this one, number 3, magenta or pink because that's what it says, and I just thought that was fun. It says solution B was hydrochloric acid. Which one of your solutions, A or B, turned pink? From your observations.
I don't remember.
Louder? B. B. It was B. So could it be hydrochloric acid?
No.
No. What does it have to be?
NaOH.
NaOH. When we did the lab and I gave the instructions to Miss Stewart, I didn't include the second side. I just gave her the instructions. So she made A and B just randomly. And so when I originally wrote it, I wrote it with a specific one in mind. So that's completely my fault, not Miss Stewart's. So the correction is just cross out B and write A, and cross out A and write B. What I did below is I rewrote the sentence correctly if you wanted to rewrite the whole thing. Solution B was sodium hydroxide, solution A was hydrochloric acid. But if you just cross out B and write A, and cross out A and write B, you'll have it. It tells you what it is. The second correction is, question number 2, I think, says, what is the approximate pH of your sample? And I just missed that because originally this lab I had written for students at Stelly Secondary School, and they live in a home with like their real parents. And this is I don't know if you've noticed, a boarding school for most people. And if I ask for samples from home, essentially only the day students show up with anything interesting, and all the boarders show up with whatever they could find in their house fridge, usually stolen from someone else. So I gave you 5 unknowns, and I rewrote the whole lab to do 5 unknowns and then forgot to change this question. So on a scale of 1 to 14, what would the approximate pH of the unknowns be? And all I'm looking for, because litmus paper and phenolphthalein is above 7, below 7, or equal 7. Acidic, basic, or neutral. I'm making sure you know what the pH scale is for acidic, basic, and neutral. So take out your sample and just put pH of the unknowns. Okay, yes?
I already answered that question.
I'll just correct it. Or add it, if you did 2 with a sample, then later on you can just write 2 rest of samples. You can also at any time, somebody asked me, can I make a table? This is science. I love tables. Give it a number and a title and go like, here you could go table 4, approximate pH of unknowns, put the unknowns, put their approximate pH, make it a proper table, done. Then you can refer to it. If you previously made a table of that in your observations, and it's table 3 where you said what pH it was, you can then say there, refer to table 3, column 4, labeled pH. The whole point of numbering and labeling them is so you can do things like that. Full sentence as a reminder, you're going to take your book away with you so you have it for prep. If you need help with any of these, you could ask now. You will get you're going to bring your lab book, so don't forget to bring it on the quiz day. You can't use someone else's, you have to use yours. In your lab book, have a copy of the procedure. You might need it. If you have lost this, I have printed new copies. Because I've been correcting it, they already have the corrections on it. So they will look a little different from the one you have, but it's still the same.
Can I get that?
Sure. Also, your book should still have the marking sheet in it. Leave that in there. Make sure those pages in the front. When you hand in the quiz, you're going to put it inside your lab book and hand it in. Because on Christmas Eve, December 24th, while I wait for Santa, my favorite activity is to light a fire, sit back in a big comfy chair, and mark labs.
Really?
Dude, yeah. So much fun.
Sounds like fun.
Or the other time is when I'm in Privet House on Thursday night, staying up till 6 a.m. to make sure everybody gets up for the bus.
Yeah.
All for the boys.
For the boys, yeah.
For the boys. Yeah.
Pretty inspiring.
I think so. Are you
Don't
bus
they
captain?
have a
I am like that.
Nice.
God,
No, in Privet we have bus captains. For every group of people that go, I designate a bus captain and make sure everybody goes with them.
oh no. For us, it's just kind of if you don't get out of it. So. If you just want to, you got it.
Well, this is why Privet's the best way.
Pardon?
This is why Privet's
That's why Privet's the best.
No.
Okay, any questions with what you're doing or
we
anything?
have a mystery.
Okay, I just realized I'm recording. I could probably stop now. You have the rest of the class to work.